do. Shoot. Let's see here. Ah, there we go. I had to check the presenter view box. Okay. All right, so first of all, I would like to thank the organizers for allowing me to share with you this work today. Most of what I'm going to describe here was published this past fall in Nature Chemical Biology and is the result of a nearly decade-long collaboration between Chad Meyer's group at the University of Minnesota, of which I am a part, and also Charlie Boone's lab at the University of Toronto, and the Yoshida and Osada labs at the Riken Institute in Japan. The primary motivation for this particular project is to gain a better general understanding of how chemical, small molecules, and biological systems interact. And the idea being that if we can find a way to rapidly functionally annotate compounds, then we can not only find new and potentially useful compounds with new modes of action, but we can also take these compounds that have good annotations and infer some general properties of these chemical and biological interactions. So this method for rapid functional annotation in our project is based in yeast. And the yeast is a great model organism. Being a eukaryote, it is a very good model for higher organism biology. It is also very genetically manipulable. And that ability allows you to create whole genome deletion collections and other mutant collections. And also the organism is so well studied that it has great annotations in ontology, such as the annotations to the gene ontology. And what I'm showing you here is for our chemical genomics experiments in yeast, we take these deletion collections that I mentioned, and we treat all of these mutants with a compound, and we look at which strains are either sensitive or resistant to that compound. And the identity of the strains that show those differential effects gives us clues as to what the compounds are doing inside of the cell. So you can think of this as a high-dimensional quantitative fingerprint of compound function. And the identity of these mutants themselves give us information, but also the nature of this profile being a fingerprint allows us to, if we have the right set of profiles, if we have the correct compendium of profiles, we can actually interpret across the genome. And what I have here on the left side is a genetic interaction network, and you will notice that conceptually, the only difference between the chemical genetic interaction profile on the right and the genetic interaction profiles on the left is the label on the rows. Here being a drug, and on the left side being another gene deletion. So when these two profiles are similar to each other, it's as if the drug is knocking out the gene. And the hypothesis that we generate that there's now evidence for is that the drug is actually inhibiting the function of that compound. So there's a theme of network integration here. And if you're not familiar with what the power of these functional profiles using deletion mutants in yeast, if you take the matrix on the left side and you compute all pairs, or you generate all pairs of mutants, and then to generate an all-by-all matrix and compute pairwise similarities on the rows, you end up with a map of the cell like this, wherein genes that have similar functions to each other cluster together at multiple levels of the hierarchy. So I'm showing you the highest level of the hierarchy here, but if you zoom in on this map in different places, you can also see that genes with even similar, very specific functions cluster next to each other. So with the idea of taking compounds, rapidly screening them, and putting them on this map, we had to do a couple of experimental tweaks to actually scale this assay up to high throughput. One of those was to make the yeast much more sensitive to chemicals. So yeast are very good at pumping chemicals out of the cells in a way that human cells are not necessarily able to. So we downregulated or deleted the genes that are responsible for that in yeast so we could screen large compound collections that have small amounts of compound in them. And we also are taking advantage of the fact that we are interpreting these profiles using the compendium of genetic interaction profiles and the fact that we do not need all of the mutants in order to recapitulate those similarities. So using an approach grounded in information theory, Ramesh Deshpande, a previous member of the Myers lab, wrote an algorithm to compress the collection of about 4,000 deletion mutants to about 300. And that's the collection that we used, which allows us, with a 10-fold reduction in the number of strains, we get a 10-fold increase in the amount of samples we can fit with the same amount of sequencing. And to show you the final step of how these experiments actually work, we take our bar-coded deletion collection, grow them in pools in competition with each other, one pool in each well of a 96-well plate with a different drug in each well. We can actually, through an indexed PCR process, combine 768 samples, so eight plates of samples into one lane of Illumina HiSeq sequencing, and it's sequencing these amplicons that have both index tag that codes for the compound and a barcode that codes for the gene. Using a pipeline called Bean Counter that I've been the primary developer on for the last 3 years, we convert the raw sequencing into chemical genetic interaction scores. And to give you an idea of how many compounds we've screened so far, we've screened about 10,000 compounds from our collaborators at the Recon, and about 3,000 more compounds that have a bit more clinical focus, both plated sets from the National Cancer Institute and some compounds with history of clinical trials, and a freely available kinase inhibitor collection from GlaxoSmithKline. And overall, we have analyzed over 50,000 conditions, screened more than 15 million chemical genetic interactions, and are nearing 2,000 compounds that we have high confidence predictions at the biological process level. To give you an idea of what these profiles look like, what I'm showing you here is a clustered heat map of the negative chemical genetic interactions for nearly 1,400 compounds that have our highest confidence biological process predictions. And I want to show you some of the functional clustering that occurs in this heat map. So the first example I will show you is we have a cluster that's driven by genes that are involved in DNA replication and repair. And the compounds that are in this cluster are known DNA damaging agents, and also excitingly many novel compounds that are showing the same chemical genetic profiles. Similarly, we have a cluster driven by one of the tubulin subunits as well as the tubulin folding factors, and this has known tubulin polymerization inhibitors, as well again as some novel compounds. Finally, we have a cluster here with known histone deacetylase inhibitors, and the genes or the mutants that drive these profiles are part of the SWARE1 chromatin remodeling complex. So looking at the clustered heat map is cool. You should do it for all large data sets, but it's not a very quantitative way to actually make predictions. So I wrote a pipeline called ZG-Target that takes the similarities between chemical genetic and genetic interaction profiles, maps them onto gene sets, and by accounting for sources of experimental variability, we end up with, for each compound, a set of, in our case, Go biological process predictions. So these processes we predict are perturbed by these compounds in the cell and with associated false discovery rate. And that's how we draw our high confidence threshold. To be a bit more concrete about what happens here, I will show you an example of a compound, NPD4142. Its top process prediction was mRNA transport. The chemical genetic interaction profile is here, showing with blue being the negative interactions and yellow being the positives. On the left, we have genes that are the genetic interaction profiles for the genes that map to this particular Go term. And on the right side, because the heat map is a little damped in the dynamic range, we show you more quantitatively where these profiles are actually lining up. And what you can see here is the strongest concordance is driven by genes that are involved in mRNA stability and the nuclear pore, which is involved in transporting the mRNA out of the cell, or excuse me, out of the nucleus. We validated our biological process predictions for compounds that we know what they do in the yeast cell. And as on this page, I don't necessarily need you to look at everything. There's only a couple examples on this of things that don't line up. So we have a more quantitative description of this too, but that's not for this presentation. But we also wanted to validate novel compounds as well. And what I'm showing you here on the left side is a compound NPD 2784 that was predicted to inhibit the tubulin complex assembly, was the Go term. And we took it into a mammalian in vitro tubulin polymerization assay and showed that it indeed does inhibit this polymerization. We then took the rest of the compounds that had the highest level of confidence predictions to this term as well and showed that when compared to a randomly selected set of compounds that still had high confidence predictions, just not to this particular neighborhood of the cell, that we are able to better than random predict inhibition of tubulin polymerization in this in vitro cross-species assay. So this is cool because we actually predicted the activity we're predicting spans into the mammalian space. Now, with rapidly annotating compound libraries, we can actually begin to characterize compounds at the library level, possibly for deciding which libraries would be good to use in the future for a certain screening campaign. And I wanted to just point out, before we even get into the functional characterization, we can just look at which libraries were better at producing hits. And interestingly, the largest collection from the Recon had the lowest hit rate, and the collection that had the highest hit rate was the kinase inhibitors from GSK, quite possibly because these were further along in the optimization pipeline, so they were better they get into cells better already. And they also, kinase inhibitors are kind of known for being promiscuous even within a species, so it's very possible they would be promiscuous across species as well. Now on to some of the functional annotations of these compounds and at the library level. What I can show you here is how we put these compounds on the map. First of all, if we want to look at individual compounds, we can look at all of the genes that had similarity. So in this case, tunicamycin's most similar genes are mostly showing up in glycosylation, and tunicamycin is a glycosylation inhibitor. Same with MMS, which is a DNA damaging agent. It lights up the DNA replication and repair regions of the cell. Then we can, for each compound, we can change it into one node on the map based on the top gene that drove its top process prediction. And we can start to see where these libraries fall on the map. So the Recon NP depot looks reasonably diverse, although you can start to see that maybe there are some blind spots, such as only one hit in the ribosome biogenesis category. The Recon natural products is a smaller collection, and we can certainly see there are some places of the map that it does not light up at all. And we also have interesting, these diversity sets which were selected to be either structurally or mechanistically diverse, and we can see that it's not an even distribution across the cell. And I'll treat this more quantitatively though, because the graphs are cool, but they're not necessarily quantitative. And first of all, we can just count where all these compounds end up in terms of their annotations. So we see there are certain processes that are very highly hit by these compounds according to our predictions, and then some that are not so much. But it also, this doesn't control for what our expectation would actually be. Some of these processes might just have a lot more genes and proteins involved in them. So by comparing these proportions to, or these counts to a reasonable background, we can better characterize what the biases of these collections might be. And we can see for most of the collections we screened, we tend to hit glycosylation-related processes a lot, which may have to do with cellular stress. But we also see a depletion for things that happen near the inside of the cell, near the nucleus. So a depletion of compounds that target DNA and other transcription-related processes. And that's interesting. And we only see this flip when we look at compounds that are specifically targeted for human uses and specifically anti-cancer drugs. And I'll also point out the black regions of this map are not no annotations, it's annotations at the expected level. So we also see a lot of things happening at the expected level, showing that our functions that we're hitting are diverse. One quick point I wanted to make on, if we can go back to this particular figure, showing where these profiles have negative interactions and positive interactions, and then where they are actually lining up with genetic interactions. We globally characterized this and showed that when we're lining these two profiles up, the information content where we line these profiles up is really in the negative interactions. And not only is that mostly responsible for the predictions that we make, as you can see globally here, but it actually, in different work, is shown to be the cause of our accuracy as well. we have, and there's an interesting part of this plot, which is genes that tend to show negative chemical genetic interactions that are sensitive to chemicals, but when they show these scores, they're not ever driving biological process predictions. So these might be chemicals that are somehow specific, or excuse me, these might be mutants that are specifically sensitive to chemicals. It would not be similarly sensitive to genetic perturbations. And we have limited statistical power because of our diagnostic set, but we have some hypotheses now by looking at these genes of processes that might yield cells specifically sensitive to chemicals. So in the last minute or so, I want to update on things that are not published and the current work that is going on in the lab to really dive deep on a lot of our high confidence compounds and link them directly to protein targets. So what I've shown you today has about 10,000 compounds from the Recon, from our collaborators at the Recon, as well as some other compounds, but we've continuing with the Recon compounds. So that's what we screened in the diagnostic set. And then we've expanded to different yeast mutant collections for about 1,000 of those compounds that we have the most confidence in. And now we span non-essential and essential gene mutants. And from those results, we are also prioritizing even further in looking for both if we can generate drug-resistant mutants and if we can overexpress essential genes and find resistance to these chemicals from those specific perturbations that would also link compounds directly to the targets. And to give you a little teaser of things that we can see when we integrate all these data types, we have on the x-axis the similarity score between chemical genetic and genetic interaction profile. And then on the y-axis, we have scores for heterozygous diploid mutants, which when you see a negative score, that can actually implicate that gene's protein as the direct target. There's been a lot of work in this area by others. So what we're looking for is a bottom right, showing a really good alignment between these two data types in the predictions that we make. So I want to direct you to my colleague Hamid Savitsadeh's poster tonight, who is doing a lot of this integration work. Wrapping up here, I wanted to just expose you to the resources we made available, both software and a website as well. So the work that I talked about has involved these pipelines for processing the data and interpreting the interactions, and then Mosaic is an interactive website, as well as a data repository for downloading, for actually exploring the data set and the chemical structures and things like that. And to wrap up, I hope that I've convinced you that we've developed this really high throughput chemical genomics system that characterizes a lot of compounds and that we can integrate chemical genetic and genetic interactions on a large scale to make predictions of compound mode of action. We validated known compounds and also are expanding very much our efforts for discovering novel compounds and validating their modes of action. And we've been able to characterize from the single compound to the library level. So with that, I've described some future directions to you already, and so I may not need to go too much into that, but we are just diving deeper on the data and the predictions and the compounds. So with that, I'd like to thank all the collaborators that have been involved with this. My advisor, Chad Myers, has been great. I'm finishing up soon, and hopefully we'll push a bunch of papers out and stuff. We already had the big paper. So with that, I will take questions.
We have time for one question. Mike's in the back, or I can hand one to someone in the front. Or we can save them for the coffee break. All right. Okay, thanks, Scott.